Assalamualaikum and hi everybody. So today we are going to enter topic 4, biocatalysis. There are 3 subtopics in this topics. 4.1, properties of enzyme and mechanisms of actions. 4.2, cofactors. And 4.3, inhibitors. The learning objective for subtopic 4.1 are: students should be able to state the properties of enzymes. Students should be able to state the 6 classes of enzyme according to IUPMB. Explain how enzyme lowers the activation energy. Illustrate the mechanism of enzyme action based on induced feed models, and state the factors that affect the enzymatic reactions. So, enzymes. What is meant by enzyme? So, enzyme is a biological catalyst that speeds up the rate of reaction by lowering the activation energy. So as we know that the enzymes are made of proteins, so the monomers that make the proteins is known as amino acids. As we know, there are 4 levels of proteins, and enzymes is at tertiary level. Enzyme is a biocatalyst. Just looking at the terms, we know that bio means life, whereas the catalyst means speed up the rate of reaction. And here are the properties of enzyme. There's a lot of properties, but there are a few main one. Okay, meaning that kalau ditanya dalam soalan structure, you jawab yang main one. Okay. So what are the main one? Among them are all enzymes are globular proteins. Okay, they works by lowering the activation energy. Okay, this is very unique because enzymes are specific. What does it mean by specific? Specific enzyme is for specific substrate. Next is enzyme may catalyze reversible or irreversible reaction. Okay, but not the main properties. Next, enzyme do not destroy after the reaction, or in simple words, enzyme can be reusable. And because they can be reusable, they only required in small amount. So you hanya perlukan sedikit kerana dia boleh diguna berkali-kali. Okay. Other than that, we have enzyme that increase the rate of reaction. Okay, this one is kind of like a main properties. Enzyme activity varies with pH, temperature, substrate, and enzyme concentration. As you can see, these are the factors that affect enzyme reactions. So meaning that here, the enzyme reaction varies depending on different pH, temperature, substrate, or enzyme concentration. Okay, so however, enzymes, the rate of reaction is at maximum at optimum temperature and pH. So different enzymes have different optimum pH. Okay, for enzyme, they can be denatured when they are exposed to high temperature or extreme pH. Okay, we're talking about extremely high temperature. So they can be destroyed. But we mostly use denatured. Okay, next, enzyme works by formation of an enzyme substrate complex. This is when the enzyme and substrate binds together in the right or correct orientation, or we can say that they are very close to each other. Okay, and they also have a properties known as the active site. So the active site is actually the binding site of a substrate. So if you have an enzyme, so this is the active site, therefore the substrate will bind at the active site of the enzyme. Okay, now how are enzymes classified? They are classified based on the type of reaction they catalyze. Okay, but before that, let's have a look how are we going to name the enzyme. There are two ways of naming the enzyme. First is naming them based on the substrate they act on. If you have a substrate, for example the sucrose, the enzyme would just be sucrase, ASE. So you change the OSE into its suffix ASE. Okay, lipid, lipase, amylose, amylase, and so on. That's one way. So we have another way, which is by naming them in three parts. For let's say we have alcohol dehydrogenase. So alcohol here is the substrate. Dehydrogenase means the reaction. Okay, so dehydrogenation, and you add on a suffix ASE. Okay, same goes with the RUBP, carboxylase. RUBP would be the substrate, carboxylation would be the reactions, and ASE. Simple as that. However, these enzymes are classified based on the reaction, which based on the IUBMP. What does it stand for? International Union of Biochemistry and Molecular Biology. So they categorize or put this enzyme into classes, where there are 6 classes. Okay, I'm not going to go very details on this, but I'm just gonna explain it to you. How are they being classified? So as you can see, one of them is the oxidoreductase. So by its classes, you know that they consist of oxidation and reduction process. Okay, so basically oxidoreductase is a class of enzyme that catalyze redox reaction, and examples is oxidases. Okay, then we have transferases, is where they catalyze the transfer of functional group from one substance to another. Okay, should be group here, functional group. And then we have hydrolases. They catalyze hydrolysis or any to hydrolyze bond by addition of water molecules. Then we have lysis. It will catalyze the breaking of chemical bonds without addition of water. Okay, that's what we say other than hydrolysis. Then we have isomerases, rearrange groups within a molecule. So in a simple way, they convert one isomer to another. And then we have ligases, which catalyze the joining of two molecules by forming a new chemical bond. So this covalent bond between two molecules use energy from the breakdown of ATP. Okay, so as where the process use ATP as its energy source to join a bond, that would be DNA ligase. This one we know them in previous semester. Okay, so the examples given, as you can see, some of them you have heard before, but some of them you might find it in the topics after this. Okay, when you understand the reaction, then whenever you encounter with an examples of enzyme, then you will know how the reaction occur, like what type of reaction that involve that particular enzymes. Okay, before we go into the mechanism, one of the important things you guys need to know is the activation energy. So what is activation energy? First thing first, kita kena faham apa tu activation energy. So activation energy is the minimum energy required to break the existing bond for a chemical reaction to take place. Basically, it's like what is the minimum energy yang substrate perlu absorb untuk break the bonds in order for a reaction to take place. Okay, so it's actually absorbed by the substrate. So let's have a look at this graph. As you can see, we have the energy of the reactant, or sometimes we call it as the energy of substrate, and then we have energy of the products. Okay, so this would be the energy of products. Normally, the energy of products is lower than the reactant because they release energy. So here is the transition state. Okay, so as you can see, in order for this reaction to take place, there is a minimum energy that this substrate required for to start or to take action to take place. This arrow here indicate activation energy. Okay, so this is for you to understand activation energy. But normally in questions, this is the type of questions that will come out in exams. Okay, where you have two types of graph here. Okay, if you can see, one in black color and one in red color. Okay, as the difference is which one is with enzymes. Okay, in the presence of enzyme is the red color here, and the obvious difference is the activation energy. Okay, it is lower than the black color here. So this arrow for the course of reaction without enzyme, this arrow indicate the activation energy without enzyme. Okay, as for the red graph, this would be the activation energy with enzyme. And in conclusion, if you can see that with the presence of enzyme, it lowers the activation energy. So you should know how to differentiate these two graphs. And what does this arrow represent? Okay, so in order to test your understanding, let's have a look at the question in your worksheet. Question number 4. Okay, I have all the labeling changed to alphabets, so you can identify all those. Okay, we start with question A to identify P to S. Okay, first thing, bila you nampak graph ni, you bezakan yang mana satu with enzyme, which one is without enzyme. And paling obvious is you look at the transition state here lah. Okay, tapi you look at the peak of the graph. So kalau dia tinggi, this would be without enzyme. Ni with enzyme. So once you dah identify the graph, you look at the arrows here. Okay, this the energy required. So the activation energy untuk Q represent the activation energy without enzyme. And R is the activation energy with enzyme. Okay, now kita tengok P dan S. So P dan S adalah energy. Okay, amount of energy at that particular state lah. So kalau P ni adalah sebelum reaction berlaku, so P adalah substrate, dan S adalah P adalah energy of substrate. Kalau you nampak reactant pun tak salah sebenarnya. So yang ni adalah energy of product. Okay. Now let's have a look at this one. Identify P2S. Okay, B, kita tengok soalan B. What is meant by activation energy? Then you just jawablah macam tadi The minimum energy required for a chemical reaction to take place, or required by the substrate to break bonds in order for a chemical reaction to take place. So how do enzymes speed up the reaction? How do they speed up? By lowering the activation energy. Okay, next, name the region of an enzyme in which substrate binds to. So if you have a substrate, an enzyme, okay, there is a site where a substrate binds to. So this site, we call them as the active site. Okay, now question E. But before that, I'm just going to show you the answer for the figure P to S, so you can refer it to this diagram. Okay, now question F, explain how the active site or the formation of enzyme substrate complex can lower the activation energy. Ni sangat penting. Macam mana dia boleh mengurangkan activation energy? So first ni apa tu enzyme substrate complex? So if you have an enzyme and substrate separately, okay, and then reaction happen, first thing, the substrate must bind to the enzyme. So when they are binded together, that they will form what we call as the enzyme substrate complex. So maksudnya kalau you ada enzyme, then you ada substrate. So bila enzyme dan substrate binds together, this is what we call as the enzyme substrate complex. Okay, so in order to explain these questions, you need to know what happened actually when the substrate are with the enzymes, or what happened when this complex form. Okay, basically this complex is to bring substrate together in a right direction or orientation. Okay, orientation. Bring the substrate together in right orientation. Okay, so when they are in a right orientation, sometimes kita kata closer to each other. Okay, next thing, what they do is this complex will facilitate. As we know, enzyme akan break the bond. So this complex is actually the one facilitate in breakage of all bonds. Then they facilitate in formation of new one. Okay, not to worry, here's the answer for that. Alright, now we are going to look at the enzyme mechanisms of action, induced feed models. Okay, so you guys kat sekolah dulu dah belajar tentang lock and key model. So I'm not going to explain it in details, okay? But I'm going to explain details on the induced feed model. This is new to you, and there is a difference than the one you have learned before. Okay, So a bit of introduction. Induced feed model was introduced by or proposed by Koshland in 1959. So he suggested that the active site is flexible, or they are not exactly complementary to the shape of substrate. Okay, so it's very important for you to mention not exactly complementary. Okay, so kalau you tengok, untuk memahami, kita tengok figure ni, kita ada active site dan this is substrate. Kalau you tengok, memang dia tak compatible. It looks like they have different shape or they are not complementary. But then what happened? The substrate will bind to the active site. The binding of substrate ini akan induce a slight change in the active site to make it complementary. Okay, at first they are not exactly complementary, but the binding akan cause them to be complementary. Okay, this diagram here shows the induced feed model. Okay, so they start off with the enzyme and substrate, and you can see that the active site of enzyme is not exactly complementary to the substrate. Okay, then what happened next is when the substrate bind to the enzyme, this causes conformational change. Okay, there's a slight change in the shape of active site to make the substrate bind to the active sites or makes fully complementary. Okay, not exactly complementary, become fully complementary to the substrate. Okay, then barulah enzyme substrate complex form. Okay, then barulah the enzyme substrate complex form. Then what happened? Reaction take place, product form and release. Last but not least, what happened to the enzymes once the product release? They akan return to its original conformation. Okay, they can return to its original conformation. Okay, so this is all the sentences. You can use this to fill in the essay in the worksheet. Okay, cuma madam tukarkan dalam bentuk numbering. Okay, according to the induced feed hypothesis, the active site is rigid and it's not exactly. Oh, sorry, it's not rigid. It should be flexible. Okay, can change it to flexible. Rigid ni adalah yang block and key models. My bad. Tapi you can fill in flexible and not exactly complementary to shear substrate. And the substrate binds to the active site. The binding induces slight change in the shape of active site. Now the active site becomes fully complementary with the substrate. Formation of enzyme substrate complex. Reaction take place. Products are formed and released. And last, the active site returns to its original conformation. So here is a diagram. Illustration of the induced feed model from Campbell's. You can take time to read this. It is also available in your diagram book. Okay, now this is a animated video, a small one, just showing that at the beginning the active site is not fully complementary to the substrate. But once the substrate bind, this cause a slight change. Okay, you nampak macam active site tu kind of like hugging the substrate kan. Dia maksudnya dekat sini berlaku perubahan dalam bentuk active site tu. Okay, this is a worksheet question. You can refer to your worksheet. Okay, it's exactly the same illustration macam tadi, just madam change a bit of it. Okay, so identify P to T. So the P is of course the enzyme, R is the substrate. Okay, and we know that the substrate bind to the enzyme, so Q would be the active site. And then we have S. S is when substrate is close to the enzyme, they bind in the right orientation. So this is what we call as the enzyme substrate complex. Okay, then we know that reaction take place and T form and release. So T would be the product. You write in full lah. Okay, name the hypothesis shown by the model above. So kalau you kadang-kadang mungkin you confuse dengan lock and key, so you check je. Kalau dia tak exactly complementary to each other, then this would be the induced fit model. Kadang-kadang soalan cakap hypothesis, okay, boleh juga jawab hypothesis, tapi lebih tepat dalam model lah. Okay, explain why amylase is unable to catalyze the breakdown of a protein into amino acid. So ni soalan application. Dia kata ada satu enzim amylase. Amylase ni enzim yang akan react dengan protein. So protein di sini adalah substrate, amylase adalah enzim. Sepatutnya substrate ini dipecahkan ke amino acid dia lah, which is the product. However, dia kata enzim ini tidak boleh, unable to catalyze. Why? Ni you're going to relate dengan properties of enzyme. Okay? So kalau based on this question, simple je sebenarnya. Kita tahu specific enzyme react to specific substrate. So kemungkinan besar enzim ini adalah enzim yang salah. Okay? Tapi ada kita jawab dia enzim yang salah? Tak, kita kena jawab one by one. So cara nak jawab soalan ni, you kena tackle properties dulu. So properties yang kita nak tackle adalah enzyme are specific. Okay, specific in terms of what? Is it like the shape or what? So specific dekat sini we are talking about the active site. So the active site has specific shape. You explain properties. Now baru kita tackle question. Okay, kita nak tackle adalah amylase tu. So kemungkinan dekat sini active site of amylase, okay, don't worry, jawapan tu akan ada kat belakang, sebenarnya is not complementary. Kita tahu substrate kan, tapi kita yang substrate kita use from the question protein. Okay, complement to the shape of protein. So what happen when they are not complementary? The substrate cannot bind to the active site. So last point, the protein cannot bind to the active site of amylase. Okay, so remember, you tackle properties dulu, you explain the properties, then you relate to the questions.